This is the story of how a team of researchers is using gut microbes to make products out of broccoli that can improve our health. Our story takes place at several universities, because the research we do on broccoli sprouts and gut microbes is a collaboration between experts in nutrition, biochemistry, microbiology, and health. In fact, this research is only possible because we each bring our extensive knowledge to problem solve together. The nutrition and biochemistry parts are led by the Li and Zhang Labs at SUNY Binghamton, and the microbial parts are being led by the Ishaq Lab at the University of Maine. Collectively, we are researching how our gut microbes can be used to make things out of inactive parts of our diet, which can then be used to help us. So, why are we focusing on broccoli? There are many other compounds in fruits and veggies which can be helpful aside from dietary fiber. The compounds we are particularly interested in are called glucosinolates, which contain glucose and sulfur, along with other components, and which we can't use on our own without some help. Some examples of cruciferous vegetables include broccoli, kale, cauliflower, cabbage, bok choy, and arugula. But there are many others. They all contain at least one type of glucosinolate, and the sulfur in it gives these vegetables their classic flavor and scent. Cruciferous vegetables are healthy because they provide a lot of different compounds, some of which our body can use directly. Because microbes excel at biochemistry, they can help us transform inactive compounds into something we can use. The ones that we're focusing on can be used as antioxidants and anti-inflammatories. The specific type of glucosinolate compound we are interested in is called glucoraphanin, and it gets released from vesicles in broccoli cells when they are damaged because of insect herbivory. Broccoli and these veggies also contain an enzyme called myrosinase, and when the plant is damaged or gets eaten, the enzyme comes into contact with the glucoraphanin precursor and can convert into sulforaphane to help protect the broccoli from insects. Glucoraphanin is higher in broccoli than other brassica veggies, but it is higher in broccoli seeds and sprouts than mature broccoli. Our focus is on inflammatory bowel disease, including two major presentations of it, ulcerative colitis and Crohn's disease. We are interested in how dietary components can be used to reduce inflammation and act palliatively with other treatments to resolve symptoms. What's cool is that sulforaphane acts as an anti-inflammatory compound that the human or animal host can use. If you eat raw broccoli sprouts, you'll absorb that 15% SFN in the stomach. Unfortunately, there's another enzyme in broccoli that usually steps in and makes a different end product, which is inactive to us. However, when cooked, said enzyme is inactivated so that glucoraphanin remains in its original state in the food. Mammals don't have the enzymes to do anything with the precursor, but there are some microbes in the gut that can take glucoraphanin precursor and convert it to sulforaphane for us. If you feed steamed broccoli sprouts to mice, we find high concentrations of sulforaphane only in the colon tissues, indicated by the black bars on the graph. This implies that the microbes which can do the conversion may be located in the colon. This is great news because usually if you want to resolve inflammation in people with IBD, the colon is where symptoms often appear. Here are some of the questions we are asking for our research. Does the specific location of microbial communities in the gut dictate the potential benefits of diet-host microbial interactions? Will certain physiological conditions change gut ecosystems and prevent benefits? Can we use location-specific microbes to target therapeutic effects? Here are some of the photos from the Ishaq Lab. We use anaerobic chambers to keep oxygen out and grow gut bacteria inside. We sequence bacterial DNA to identify which bacteria are present and which samples are similar. We then amplify specific parts of bacterial DNA to count how many genes they have that make enzymes to process glucosinolates. I would like to end by acknowledging the contributions of a large team related to this ongoing work. Thank you for attending this presentation. For more information, you can scan the QR code or contact Sue Ishaq using one of the methods below.